these are conducted by a toolkit of different enzymes. So these restriction enzymes, or which are also called as restriction endonucleases, these are used as a molecular scissors. So these restriction endonucleases, these are called as molecular scissors. Then this enzyme, which is called as DNA ligase, it is used to connect two pieces of DNA together. And there are variety of different types of enzymes or additional enzymes that modify DNA, and these are used to facilitate this genetic engineering process. So we will see these DNA modifying enzymes. So the restriction enzymes, or which are called as molecular scissors, and that DNA ligases, they represent the cutting and joining functions in DNA manipulation process. So basically these restriction enzymes, these are used to cut the DNA molecules at specific spaces, and these DNA ligases, these are used to join that DNA fragments. And all other enzymes which are involved in genetic engineering, they are included in a broad category of enzymes which are called as DNA modifying enzymes. And these enzymes, these are involved in the degradation, synthesis and alteration of the nucleic acids. So here, so these are the cutting, modifying and joining DNA molecules. And to perform these three steps or three processes, that is cutting, modifying and joining, it requires first, there are different types of enzymes here. Then these enzymes, these are purified from wide range of cell types. Whatever these enzymes are here, these are purified from wide range of or different cell types, and these are available from commercial suppliers also. Then later on, these enzymes, these are catalytic protein molecules, because these are enzymes and we know that all enzymes are protein in nature. So there are three main groups of these enzymes which are used for cutting, modifying and joining of DNA, and that three types of enzymes includes, the first one is restriction endonucleases, then second one is DNA ligase, and third one is here, that is DNA modifying enzymes. These restriction endonucleases, they cut down this DNA molecule at defined recognition sites. So at particular sites, these restriction endonucleases, or which are called as molecular scissors, they cut down that DNA molecule, and that sites where that restriction endonucleases cut that DNA molecule, it is called as recognition site. Then these recognition sites or these enzymes, these are used for generation of DNA fragments. So these restriction endonucleases, these are used for the making of small fragments of DNA molecule. Then these are used for the restriction mapping of that DNA. Then later on, these DNA modifying enzymes, which is the second type of enzymes which are used in cutting, modifying and joining DNA molecules. So these are the DNA modifying enzymes. As the name suggests here, they modifies that DNA molecules. And there are three different types of DNA modifying enzymes. One is, that is nucleases. The first enzyme is here, these are called as nucleases. Second one is here polymerases, and third one is here, that is called as enzymes that act on terminus. These nucleases, these are of two different types. One is endonucleases and second one is exonucleases. Then these endonucleases, these are restriction enzymes or DNase 1, and exonucleases, this includes BAL31 and EX43. Then polymerases includes DNA polymerases, then Klenov fragments and reverse transcriptase. So these are the enzymes here which are called as polymerase enzymes, that is DNA polymerase. Then second one is Klenow fragments, and another one is here, that is reverse transcriptase. Then third types of DNA modifying enzymes, these are called as enzymes that act on terminus, and that includes alkaline phosphatase. So the enzymes which act on terminus, these are alkaline phosphatase enzyme. Then second one is here polynucleotide kinase enzyme, and third one is here, that is called as terminal transferase enzyme. Then the next or the third type of enzymes used in cutting, modifying and joining of DNA, that is called as DNA ligase. And this enzyme, it joins the DNA molecules or fragments of DNA molecules by phosphodiester linkage, and they generate recombinant DNA. So they modify they joins DNA molecules by forming this phosphodiester bond between nucleotides or phosphodiester linkage, and these DNA ligase, they are used to generate recombinant DNA molecule. So in one DNA molecule we can insert the fragment of another gene, and these genes, they are connected with each other or they are joined with each other by using this DNA ligase enzymes. So friends, we have seen here different types of cutting, modifying and joining of DNA and that enzymes here. So in next lecture we will see about the types of modifying enzymes in details.